Hello, thank you for stopping by my poster. I'm Tejeshwini Rajaram. I'm a third year student pursuing a BE in Printing and Packaging Technology in Anna University in India. I am here to present on investigation of mechanism and effectiveness of metal nanoparticles in self-sterilizing packaging. So let's move on into the poster. Packaging materials play an important role in extending the shelf life of food products. They extend the shelf life and they preserve the quality of food as well. They do so by protecting food from water vapor, oxygen, and carbon dioxide. When we talk about antimicrobial or self-sterilizing packaging, they do so by combating with microbes. Microbial contamination is the main problem when food safety and food quality are concerned. Now, antimicrobial or self-sterilizing packaging materials are fabricated in such a way that the antimicrobial agents are either blended along with the packaging material or they are given as a coating over the packaging material. Now, let us look at the mechanism. On interaction with the bacterial cell membrane, the metal nanoparticle first affects the membrane integrity. Once the membrane integrity has been attacked, it leads to induced oxidative stress. Induced oxidative stress disrupts enzymes, proteins, and finally the DNA. And when DNA is damaged, it causes cell death. Another mechanism by which the metal nanoparticles attack bacterial cells are through reactive oxygen species. Now, nanoparticles can attack the microorganisms without any direct contact, and this is how reactive oxygen species comes into the picture. The reactive oxygen species in turn migrates from the surface of the packaging material and it reacts with the bacterial cell. And this reaction causes oxidation of lipids, proteins, and finally degradation of DNA. Now, I'd like to share what I've learned about different metal nanoparticles. Starting off with silver nanoparticles. Silver nanoparticles have proven to be potentially safer to use. The properties can be enhanced by adding additives to them. Also, by combining two metal nanoparticles together, the properties enhanced as well. For example, from one of the previous studies where copper metal nanoparticle and silver metal nanoparticle was combined and reinforced in LLDPA films, they showed an appreciable bactericidal activity against certain foodborne pathogens like Listeria monocytogenes, Salmonella typhimurium, and Campylobacter jejuni. These bacteria were found on raw chicken, and from this study it was also inferred that the raw chicken shelf life was also increased to almost 21 days. Now let us look at zinc oxide nanoparticles. Zinc oxide nanoparticles enhance the mechanical properties, thermal stability, and crystallinity of pristine films. Also, it is important to note that they did not alter the structure of these pristine films. They did not introduce porosity. We can tell that they did not introduce porosity from one of the results from a previously conducted study. It was noted that the oxygen transmission rate of PVA films before and after introducing zinc oxide nanoparticles remained same. So the porosity that we can control, that porosity has not been altered. But then it is also important to note that the oxygen transmission rate decreased to a minute level when zinc oxide concentration was increased. When magnesium oxide nanoparticles are taken into consideration, they too enhance the mechanical properties. But then they blocked the cracks on the surface that were caused due to an deforming force. But this too did not last forever. When the concentration of these magnesium oxide nanoparticles exceeded 3% weight, it had a negative impact on the mechanical properties. And this was because when the concentration exceeded 3%, it caused agglomeration of the nanoparticles. The nanoparticles joined to each other to form large masses, and these large masses interfered with the structure of the pristine films, and this decreased the mechanical strength of the films. And when we look at the antibacterial or antimicrobial action of magnesium oxide nanoparticle, it was noted that E. coli showed more resistance to magnesium oxide nanoparticles because the MIC and MBC values for E. coli were greater than Bacillus subtilis. Gold nanoparticles showed that there was a significant increase in the tensile strength and Young's modulus. But then it was also reported that the elongation at break was decreased. When they were tested for the antimicrobial activity, gold nanoparticles showed good antimicrobial activity against Salmonella bacteria. Salmonella bacteria is a foodborne pathogen. And the minimum inhibitory concentration of the gold nanoparticles increased on small scale with the increase in their size. With respect to titanium dioxide nanoparticles, it was observed that the LDPE films with silver and titanium dioxide and LDPE films with silver and copper and titanium dioxide nanocomposite films exhibited the maximum antibacterial protection to tilapia, which is a fish, when the concentration of silver, copper and titanium dioxide nanoparticles were 2.5, 2.5 and 5% respectively. From one of the previously done studies, it was noted that ferrous oxide nanoparticles along with PVP films or polyethylene glycol films increase the antiviral properties. So this is one of the unique results that I observed while reading so many research papers. And then the copper nanoparticles along with carbon and PLA nanocomposite films showed good antibacterial action against Pseudomonas species. Thus, to conclude, self-sterilizing packaging are widely used in areas where pathogenic microbes are present. This seems to be a clever solution to any pandemic breakdown. Almost all the metal nanoparticles follow the similar mechanism of producing ROS, or they come in direct contact with the cell membrane. Now, silver nanoparticles are extensively used in commercial packaging and have increased the shelf life of food products. And works of literature on gold nanoparticles in this subject are very few, which might be because of its high cost. And copper reinforced cellulose nanocomposite packaging materials that were developed showed good biocidal activity against E. coli. Thus, it is convincing that metal nanoparticles prove to be better antibacterial or antimicrobial agents. And they can also be used in food packaging to produce self-sterilizing packaging and decrease the use of chemical preservatives in the coming future. Thank you.